My name is Hae Lee from Samsung Medical Center. I'm going to report back about the cancer breakout sessions. Instead of discussing a lot of different general ideas, we focused on more specific issues in HCA-Asia cancer field. So we brought up three actually Asian, specific to Asian cancer community, or actually just a cancer field. So one thing was, the one important topic was what is unique that we can provide as part of HCA Asia to HCA Global. And from this topic, we thought about unique patient configuration, very unique in Asian countries, like breast, liver, gastric cancer. And we can start incorporating data for those cancer types, and then contribute to the global cancer field. And this is important because there's, of course, diversity. And since we don't have as serious issues of getting tissue samples for cancer, it's really difficult to get normal in Asian countries because of many reasons, but we have plenty of cancer tissues, so probably we can start this pan-cancer or pan-institutional studies and expand it to persuade the funding agencies or government to feed the fund to the matched normal or even real normal cases. So we can kind of expand this cancer area into the real normal data set. So, so we agreed on working on white paper on this project as the other breakout session. We haven't got the name, so if any volunteers, we can probably start writing the white paper on this issue as well. So again, the last ones, like we can start from the cancer field and then go to the normal. So it'll be actually easier to get funding and persuade the funding agency in Asian countries. Another important issue and unique in a way is the longitudinal sample collection, because it's also for other disease consortia, but for the cancer field, it's really important to look at the treatment resistance. So issues involved with the longitudinal sample collection was a bit unique in cancer. And there were actually two points raised. One was those access to clinical trials and influencing industry partners. We can do the clinical trials and funding for the those longitudinal studies in the Asian countries. The second one was, I put it in different slide, was actually preserving the samples for a long term, so we can have this treatment resistance. That's like 2 years, 3 years, even longer sometimes, and we have to come up with the best sampling strategies, and probably we can share tactics on it. There are many ways to do it, just storing the samples and waiting for a long time, or just generating a lot of data by multiplexing. So there are like a more unique situations we need for the cancer studies for the multiplexing because of those longitudinal sampling issues. And like other normal or disease settings, extensive metadata required. So cancer may not be single disease. So if you look at like a different aspect, even stage of cancer, so it could be very different, different aspects. So the extensive metadata required that we can come up with some common form for the required clinical information that we can share for the pan-cancer analysis. So these are the issues came up with more discussions in the session, and others are like about a bit redundant. So we discussed this pan-cancer analysis. I just talked about it, and multimodal analysis for cancer, longitudinal studies. I discussed in the first slide. So that's redundant. Let's go to the next one. We discussed about this ISIC panel in cancer specific way. So there are a lot of reagents targeting the immune cells in like a commercial source, but not as many for the cancer cells. And probably if we pursue this with and talk to the companies to make more cancer cell specific antibodies, probably it will boost the research area. So we talked about this possibility. Extensive metadata and the spatial gene expression data, whether that's gonna have additional layer of information for the cancer field. Of course, yes, there are many groups who's looking at the spatial distribution of cancer cells or immune cells together. And again, for the diversity-wise, it's an important issue because this spatial gene expression actually relates to the pathology data. So if you have some unique pathology like a breast cancer in Asian women, you can diagnose better, and there are a lot of development of this digital pathology. So if we don't contribute enough of data, we may be like excluded out from those new technologies. So probably this is another field that we have to consider very actively to put an input from different Asian population. And the last one's use of preclinical models. It's more of like a research-oriented issue, so there's a need for the preclinical models, but at this point, it's not really going to be a part of this HCA-Asia cancer field yet, just not yet. So these are the issues that we have discussed, and if I have missed anything else, someone can raise a hand and point it out. Any opinions? Different points? Okay, go ahead.
See, really nice to see that you discussed some concrete project, because actually it's a very nice segue into the next session where we're planning what can HCA-Asia do beyond the existing projects, what new projects can we launch. Question, I really like the idea of comparing cancer types across ethnicities or population groups. How easy or hard is it, again, going back to the issues Christian raised about harmonization of the precise location in the organ where you sample and things like that. So if in cancer you want to say you had to compare the same stage, presumably compare the same grade, maybe not stage, but at least compare the same grade of tumor, same anatomical location, and same everything else. But in two different countries, and you have limited control over the surgeons and how they do their job.
Right.
So how does that work, and getting rid of all those confounding factors?
That issue brought out in the meeting as well. We can try to control like an afterwards process. Once we get the sample, probably we can try to coordinate it and do a good job. But then before getting the sample, I mean, surgeons different, there are factors we cannot control. So what we suggest was that with all those confounding factors, we can have this multi-center data collection and do the analysis. And if we do find some difference, we can actually go back and use different platform like conventional immunohistochemistry, and it will be actually easier to look at the slide and see the difference. But we can pinpoint what are the points that are suspected different between like different locations, those. So with all the limitations, I think it is better to start the project, and it's important that we know what are the limitations, so we don't get fooled by the differences from like batch effect, and then come up with a validation strategy.
Maybe comment on that again. Yeah, it's a really important point that you brought up, of course. I think that's the thing that we discussed, that maybe the analysis has to be done in parallel, so we share data across sites. and then do analysis individually, right, in parallel, and then look to see if we can find consistent differences on cell type, cell states between tumor and normal. Of course, this is all in the context of disease versus normal, right? And if we find those same differences, then we believe them to be. So we need to kind of do this first to kind of come up with a set of kind of ground truths, if you will, right? And those would be then more believable. But of course, there are certain things that are completely out of our control in terms of sample acquisition, surgeons, all of that, right? But yeah.
Was there a discussion on harmonizing technology? So I think we've been, especially in cancer, the RNA and even DNA profiling of the same cell could have a tremendous benefit. And there have been some talks about OneSeq, or there's now rumor that 10x may release RNA-seq with ATAC-seq in the same cell, and there are other examples of that from other platforms. So is there ways to harmonize across the consortia to gain this information?
Yeah, I mean, good point again. So I think we focused mainly on what we could do together first. But one of the things that Hio pointed out was this multimodal. So we didn't dwell on it. We went through it really fast. Yeah, number 2, right? So in terms of combining a single cell ATAC with DNA or a single ATAC with RNA, and as well as looking at the mutational profiles. In terms of, we didn't go into specific details of how best to harmonize and which platforms that we're using, but most of us are working with 10x anyway. So, but that's something that is up for discussion. But we would love to get your opinions on it as to how best to harmonize it. We were focusing mainly on what we should be doing, and then we'll figure out a way to do it.
Actually, we also discussed about infusing. Okay, I'm getting ready. Infusing cell lines. So with pick a case and infuse cell line and produce the data together. That was one idea. And since we already generate, many institutions have generated the data with their own protocols, we can again select cases that we try like a common protocol and see whether it mirrors whatever we found with other cases. So we can have those cases together.
Any other questions? All right.
How unique are Asian cancer? I guess it's a scientific question and we don't know the answer, but I guess you're going down this path saying there is, because it addresses your first question, which is what unique thing can HCA-Asia contribute? And how much do you believe that there is some unique cancer biology in Asia? There's unique prevalence, which is not exactly the same thing.
So actually, breast cancer is an easy example. So it's known that there's more young breast cancer patients in Asian women, and also in pathology data, it's notorious that the Western developed pathology doesn't work on the digital pathology doesn't work very well on the Asian cancer patient. So there are some differences, and I think depending on how we make a patient profile, we could probably capture different proportions of the patient. So probably use that information, not just the single cell information, but the patient distribution information together. So actually we have single cell data representing the unique patient population. That would be the strategy.